Good morning, ladies and gentlemen. Thank you for taking your time out to come for this presentation this morning. So we're here at the University of Ilorin Teaching Hospital Pharmacy Department, and we want to talk about drug hypersensitivity reactions, characterization and assessment in the 21st century. That's part of their clinical meeting series that's usually organized by the Department of Pharmacy of the University of Ilorin Teaching Hospital. I am Dr. Mohammed Amali, and I'm from the Department of Pharmacology and Toxicology, a proud sister of this department in the University of Ilorin Teaching Hospital. Now, we start. For today, we're going to look at the general theories of immune stimulation of small molecules, and we are going to try and also understand some of the molecular bases of immune involvement in the manifestation and pathophysiology of drug reactions that are adverse that we observe from time to time. And then we will now describe the most recent methods that are used in the evaluation of drug hypersensitivity reactions and how specific antigens or immunogens, as they're called, can be identified. And then we shall now finally round up by looking at the problems and try and recommend important steps to solve the problems that are associated with the assessment of drug hypersensitivity and characterization of these reactions, specifically within our environment, which is a resource-deprived environment. So within the next 40 minutes or thereabout, we should be able to deal with these issues. I would also like to say in advance that some of the technical aspects have been dumbed down a bit because of the general diversity of participants that are taking part in this particular presentation, so that we can all follow and understand what is going to. Some areas will not be delved into in depth, just so that we can preserve the understanding and at least leave this place with something at the end of the day. Now, what are adverse drug reactions? Now, basically, they have been defined, and this is a definition that encompasses all the previous definitions of adverse drug reactions, and they've been defined as a harmful or unpleasant reaction that is appreciable in nature, which means it's manifested, and it results from the intervention related to the use of a medicinal product. Now, the reaction that is observed will now tell you that in the future, there could be hazards attributable to this particular product. And as such, you will need to develop some prevention or specific treatment, or look at the regimen and try and alter the dose, or in the worst case scenario, withdraw the product completely. Worldwide, it's a major problem, a serious major problem, health problem worldwide, and contributes to up to 6% of hospital admission. And you find out that elderly people play a large part in this, because they make up to 75% of those that are admitted, and that's for those that are over the age of 75 years. You have about 30% of that category of people being hospitalized. Now, these adverse drug reactions manifest in different parts of the body, different organs. They are seen in the skin, the liver, the blood, the kidney, and the lungs. And the cutaneous manifestations are the most apparent, because those are the ones that are actually visible. And so you could have them ranging in severity from simple maculopapular erythema down to the more severe Stevens-Johnson syndrome or toxic epidermal necrolysis, which in some cases is also can be fatal. Now, they contribute significantly to an increase in healthcare costs, and they also extend the length of hospital stay of patients, because you find out that when a drug is now developing an adverse reaction, you find out that the individual has to recover from that adverse reaction before the individual goes. And of course, that leads to an increase in the amount of money the individual has to spend. Then you now have financial losses for the pharmaceutical companies involved in situations where the agent has to be withdrawn from the circulation completely. You find out that all the money that has gone into investment, into drug development, they lose that, and that runs into millions and millions of dollars. Now, to try and understand this, what these individuals have done is that they try to use a complex multidisciplinary approach to try and understand the pathomechanism to prevent them from occurring from time to time.
Network.
Now, before we proceed further, I would want to take you through some basics. And one of the basics is the understanding of what adverse drug reactions are and the classification, simple classification, so that we can all follow. Now, adverse drug reactions are classified into two, based primarily on their dose dependence and predictability characteristics. So, type A adverse drug reactions are known to be based on exaggerations in the drug's main effect. So, they are dose dependent, predictable, and they make up a larger amount of available ADR, adverse drug, and they're related to the pharmacokinetic properties of the drug and dose reduction of withdrawal of that particular drug would usually resolve the adverse drug reaction step. For type B adverse drug reaction, they are said to be generally idiosyncratic, although I have a problem with that term idiosyncratic because we've gotten to a stage where you can actually find out the cause of most of all these reactions. But for a layman or for general understanding, we should still use that term as idiosyncrasy. So they are said to be idiosyncratic and they provoke the host immune response as part of the response mechanism. So you find out that there's involvement of T cells, B cells, and other components of the immune system. Now, basically, the epidemiology and the impact of the adverse drug reactions now led to the development of further aspects to try and understand further. Now we were brought up with up to the concept of pharmacovigilance, right? And that is a system to monitor and report the onset and the pathways of these adverse drug reactions as they proceed. It utilizes basically clinical pharmacology, immunology, clinical medicine, toxicology, and nowadays they've started introducing pharmacogenetics into the reporting systems and monitoring system to try and understand the pathogenesis. There are a lot of issues that have arisen because of the manner that we get the information, which we shall relate to later in this presentation. Now, several classes of drugs are associated with a high incidence of these adverse drug reactions. And when we say high incidence, their incidence is higher when compared to the occurrence in other types of drugs. Basically, we have the beta-lactam antibiotics, which have the highest incidence, followed by the non-steroidal and inflammatory drugs and anti-seizure drugs, specifically carbamazepine. Now, there are other factors also that influence the generation of this adverse drug reaction that we see due to these particular agents. As we said earlier, the exaggerated effects of the drugs are mostly the reason why, and this is mostly seen as an extension of the expected pharmacological activities of these particular drugs, and that makes or takes a larger portion of the factors that influence the generation of these reactions. Other factors play a role, including genetic factors, immunological factors, some certain drug factors, which we can classify as pharmacological factors. And then we have pharmacokinetic and pharmacodynamic inter-individual variations that play a role. And there, that's where we start seeing issues of the genetic pool starting to play a role. And they could be also due to the age, environmental issues, and other sundry issues that may arise. Looking at it from the perspective that we can all relate to, those at extreme of ages are vulnerable to the onset of this reaction. So we have elderly patients, most of whom have multiple disease states and are exposed to polypharmacy. So they have multiple drugs being administered, and of course we know drug interactions and all that could lead to the development of this reaction. And then we now have infants and very young kids who have low level of organ and tissue development, and as such, drug metabolism, distribution, and other parameters are affected. And so we see the onset of this drug at that particular this particular age set. Gender also plays a role, and it has been observed in the severity and frequency of these reactions in some antiretroviral agents, and also with the way the hepatic enzyme functions. So we've seen specifically cytochrome P3A4 in Chinese women enhancing the metabolism of midazolam when you compare that to the effect in men. We also see that with the colchicine used in the management of gout that selectively affects the fertility in males but not in females. Then we look at the effect of alcohol and smoking. Now, chronic alcohol consumption affects organs. We all know that. But apart from just affecting the organs, they're able to accumulate enzymes that have the ability to transform drugs. And as such, the predicted actions of these drugs are now altered. And sometimes it could lead to damage to the liver and other accessory organs. Smoking is a potent inducer of the liver enzymes, that's the hepatic cytochrome P450, isoenzymes 1A1, 1A2, and possibly 2E1. And we all know what potent induction of hepatic cytochrome enzymes will cause. It leads to faster metabolism and eventually drug treatment failure. Then we look at ethnicity and the presence of genetic markers that could affect these particular agents. Specifically, ACE inhibitors have been known to be associated with angioedema. But this association is more prevalent in African Americans when you compare that with other ethnicities or ethnic groups. In addition, HLA genes, which we know as the human leukocyte antigen genes, have now been identified as genomic markers to several different ADRs. And specifically, we look at abacavir hypersensitivity, which has been associated with HLA B5701. Allopurinol is also associated with the HLA marker B1301, and we have a few others that are associated now that we found to realize that are associated with these HLA genes and markers. So currently, the ADR assessment generally depends on the use of questionnaires. And that's why I was saying earlier that when we go forward, we would be able to see where some issues can be raised with the nature of data collection with respect to pharmacovigilance. So we have assessment basically used, gotten from questionnaires. And after getting from questionnaire, if there's a clinical problem in the hospitals, you see clinicians using clinical tests to try and confirm the suspected allergies. So we have skin tests, we have the radioallergen tests, we have the CAST test, that's the cellular antigen test, and then we now have the patch testings that have been used. All these have been documented in previously written papers or conducted studies in South Africa, where 78.7% of patients on the heart regimen were found to possess one adverse reaction or the other. In 2012, in the United States, where they found out that combination therapy um, presented with serious adverse reaction, and stavudine specifically was implicated in over 90% of cases. And here at the University of Ilorin in 2017, the pharmacists here evaluated the adverse drug reactions among inpatients in their adult medical wards and found a plethora of adverse reactions to different um, agents in the teaching hospital here. Now, using questionnaires, no doubt, gives you an idea of what. So the importance, so as I was saying, most of these hypersensitivity studies were carried out, and they were carried out using questionnaires. And of course, using questionnaires, you would be able to compare epidemiological data from different patients. But the problem is that the molecular aspects of these reactions will not be visible. So they will not be unraveled. And you cannot understate the importance of this, because each individual has his own predisposition to the drug that he's taking, specifically or particularly when you consider the type B adverse drug reaction. So you find out that while you will say, of course, there's an adverse drug reaction, you cannot get to the root cause of that adverse drug reaction. And that's the first point. Secondly, questionnaires are prone to some form of underreporting, in the sense that, or overreporting, because you find out that some individuals will present signs that may be mistaken as adverse drug reactions, but may just be due to the interaction between one or two drugs, and cannot generally be classified as hypersensitivity reactions because of the lack of the immune involvement in those scenarios. So those type of drug hypersensitivity reactions will not be observed. In some other cases, they will not even be reported as such and just be passed off as regular adverse drug reactions because of the lack of the know-how to interpret purely visible signs. As such, further in vitro studies tailored towards the understanding of the molecular basis of these reactions should be considered following the confirmation of drug hypersensitivity studies. Now, this just tries to give us an insight into the workup of assessment of drug allergy to particular agent. So here we have a drug. If you suspect that the allergy is immune-mediated or T-cell mediated, you can run the patch test, you can do the skin prick intradermal test, which most of us know or have observed once or twice when we see that done with penicillin. And then we can do the drug provocation test, which is as a last resort because they are quite risky in case the individual is already predisposed to that reaction. You don't want to generate a crisis in that particular individual. Now, we've been using adverse drug reactions and drug hypersensitivity interchangeably. But just to clarify that issue, I would want to say that drug hypersensitivity reactions are those that have an immunological etiology, basically. So that's the main difference between drug hypersensitivity reactions and hypersensitivity reaction, adverse drug reactions generally. And they make up about 1/6 of all adverse drug reactions and are classified or have been earlier classified into 4 types: type 1, type 2, type 3, and type 4. And these 4 types are classified based on the involvement of the different components of the immune system in the elicitation of the manifestations that we see from these particular drugs. So type 1 are the mildest, while type 4 are the most severe, which we try to illustrate pictorially from the second slide, where we saw maculopapular erythema and Stevens-Johnson syndrome. So Stevens-Johnson syndrome is an example of the type 4 drug hypersensitivity reaction. Now, these are the earlier reactions, but now we found out that we have some other reactions that we could also fit into these particular classifications. And so we now have pseudo-allergic reactions, we have primarily antibody-mediated reactions, and then we now have T-cell mediated reactions. And this has now been clearly delineated. We shall not look at them individually due to the lack of time, but it's just to give us an understanding of the general background leading up to the assessment and characterization that we actually want to go into. Now, as we said previously, the immune system is the system that is implicated primarily in drug hypersensitivity reactions, which means that the immune cells are the cells that facilitate this particular process. So we need to understand how these immune cells, particularly the cells that are involved in these reactions, are stimulated on a normal basis, and how we can relate this to the effect that drugs and other chemical substances can also replicate such type of stimulation. T cells recognize antigens, but they don't just recognize antigens on their own. Now, they recognize antigens through what we call specialized antigen-presenting cells, which include dendritic cells, macrophages, and B lymphocytes, and also T cells, and which are the CD4 T cells and CD8 T cells. Now, these dendritic cells and macrophages, or these APCs, capture the antigens, internalize them, process them, and present them on the surface of what we call major histocompatibility molecules. And it's on those molecules that these particular T cells can now identify these antigens and carry out their effect. In the case of a chemical or a drug, the process has not been fully elucidated yet. So you find out that most pharmaceutical agents cannot bind directly to that major histocompatibility molecule. And so what happens is that they now have to bind to a protein, and as such, when they bind the three concepts basically that have been used to explain this: haptin and prohaptin concept. Ph.D. are not delve into because of time. Okay. Now, let us go into the meat of the matter. Basically, we've said we use questionnaires. After using questionnaires, we use the available clinical test, CAS, RAS, and skin test to assess. But we now have other methods for the assessment and characterization. Since we know that drug hypersensitivity reactions are due to immune cell activity, the immune cell is now the basic denominator there. So the first thing we do is we can look at evaluation of lymphocyte proliferation. We know that during an infection, there's an increase in the number of lymphocytes specific for a target pathogen. And so this principle has been used by instituting proliferation on cells using particular agents, and then testing for the amount of proliferation in a test called lymphocyte transformation test. And so we have a drug metabolite or a drug protein, whatever can stimulate the whatever can bind to the antigen presenting cell is administered. The drug specific T cell comes in contact with the antigen presenting cell, and there's proliferation, clonal expansion, and then it can now be quantified. And then we can now see if the drug is able to elicit stimulation on the particular cells in question. The idea being that if a drug is really responsible for an adverse drug reaction or hypersensitivity reaction, you would see proliferation. In instances where they are not, you will see a situation where there will be no proliferation of those cells at all. So this is just a schematic showing how the blood samples are collected, how they are prepared, and the classes of patients that blood samples are collected for this particular test, so that you can compare the proliferation in hypersensitivity controls. What is generated at the end of the day is what is called the simulation index. And a simulation index greater than 2 is referred to. Look at this graph. We could see a patient that was diagnosed for drug hypersensitivity. He was taking a plethora of different drugs, and he was now tested with some of the drugs he was taking and some others as standards to find out if there was a non-specific activation of his particular condition or the activation was specific. And we can see from the data there that the individual was responsive to only tazobactam. And as such, tazobactam was identified as being responsible for the hypersensitivity observed. Another example that can be used to characterize is what is called the enzyme-linked Immunospot assay. Now, for this particular assay, the cells are incubated with immunogens or the suspected causative drug, and they now produce antibodies. And the number of cells that are expressing these antibodies can now be detected using a particular equipment called an ELISpot machine. And it's said to be the gold standard of detecting hypersensitivity. Mm-hmm. This assay is usually run in tandem with the lymphocyte transformation test. So in this screen, you can see cephalosporin hypersensitivity trying to be diagnosed. And so what happens is on the top panel here, you can see graphs that show the stimulation index from the lymphocyte transformation test. And you now have figures that show the number of cells for each particular dose. And of course, we'll also have a cutoff point for that, which is the doubling of the this is specifically for the detection of circulating antibodies, cytokines, and for antigens that are secreted from cells. And so what happens is when these cells are put in, when they come in contact with this particular immunogenic agents, they release antibodies into the system. Usually this is carried out in an in vitro system, in an isolated culture, and is released into the cell supernatants. From these culture supernatants, we can measure the levels of these circulating antibodies and be able to determine whether they are above standard, above normal, or below the normal levels, which is an indication of hypersensitivity, can be used as an indication of hypersensitivity states. Now, the ELISA is a very simple method and can be run practically anywhere. So at the bottom here, we just have a small explanation that the wells are coated with antigen and incubated. Detection is via specific antibodies and substrates, and it has currently been modified to detect the presence of cytokines. These are the basic principles of this. Then we could also use flow cytometry. Now, flow cytometry is I use a flow cytometer to determine the size and the phenotype. So size and function, not necessarily function, but size and phenotype of the different cells. And basically, we use carboxyl diacetate succinidamide ester. So it's called CFSC, it's called the CFSC stain. That's the one that's predominantly used to measure the lymphocyte proliferation by flow cytometry. So it's one of the most widely utilized assays. It's capable of identifying the individual cells that have undergone multiple rounds of cell division, because it looks at them at a cellular level, each cell on its own. So it's able to look at each individual cell. And then different studies have also shown that the reduction in fluorescence, because what happens is this particular ester will fluoresce at the end of the day, and it's the fluorescence from each of these cells that is now picked up, and that value, the intensity of that fluorescence, is what is recorded. And what studies have demonstrated is that when that fluorescence reduces, it's due to cell division. So you find out that if a particular agent is able to reduce the fluorescence attributable to CFSC, then it gives us an indication that that particular agent is responsible for proliferation and could be responsible for the hypersensitivity reactions that we see. Currently, we still use CFSC in the tracking of lymphocytes. Then, last but not the least, we can also generate T-cell clones and B-cell clones. So cell clones as a tool for studying hypersensitivity. Now, nowadays it's very important, one of the most important. And as it is a bit difficult for us in these parts of the world, developing world, to carry out this particular kind of study. But currently, we have institutions that are keening into this and industries that are keening into this, because what this does is that it now gives us an ability to actually predict the onset of these hypersensitivity reactions without even the reactions being present at all. Okay. Now, up to now, what was done was that the T cells have previously been characterized. So, for example, if you get a patient that has an adverse drug reaction, the cells are obtained from the particular patient, and once the patient is confirmed to be hypersensitive, each particular cell is cloned. So you could have more than one cell going into millions of cells that are identical. And so you now have a store of cells that you can be using for your studies. This has been done for carbamazepine, lamotrigine, phenylenedione, and for a lot of the beta-lactam. So we have drug-specific T lymphocytes for all these particular drugs. Now, these clones were generated and tested for specificity, phenotype, and also for functionality. And once they were tested for all that, they are now kept as a pool. Now, they are also tested for their genetic profile. So the HLA molecules that these individuals express are also included in the amount of data that's obtained from these particular studies. And that's because you would want to have a bank of patients that express such a HLA, so that if you have a particular drug that is causing a drug hypersensitivity reaction in a particular group of people that express a particular HLA, you will be able to test it experimentally in a bank to find out if it is actually a general problem or it is just based on that individual's own variation or variability. Okay. Now, currently, studies have shown from our colleagues in the University of Liverpool, currently, this is one of the most recent studies, they have been able to prime, and when they say prime, they have been able to obtain cells that from naive individuals that have never seen the drug before, individuals that have never had an adverse drug reaction before. They've been able to obtain their cells, obtain their T cells, and make them now reactive to particular drugs. The most successful has been priming to Bandrowski's base, which is a dye that's used a lot, and it has been found to cause adverse drug reactions. Although it has been a bit difficult to prime, have been successful. So what this means is that if you have a drug in development, you don't necessarily need to take your drug into the market to see if it will have adverse drug reaction before testing it for adverse drug reaction. Once you have done your molecular analysis using your immune cells to identify what the cause of these adverse drug reactions, the likely cause could be, and the manifestations molecularly, product against a panel of cells, or if you have a bank of cells, and look out for those manifestations that usually present in the onset of the hypersensitivity reactions. And that has currently been saving industrialists or drug manufacturers billions, because they're able to predict to a certain extent that some particular drugs would cause reactions, and as such pull them out early, land into trouble at the end of the day. Now that brings us to the issues surrounding all this. We now know that the understanding of molecular events in individuals suspected or confirmed with drug hypersensitivity reactions is critical, and this is because we will need it to develop proper therapeutic for these, so that is not conducted on this part of the globe, so on the dark continent. By the time we look at the studies that are being conducted, if you are even writing a paper or you are trying to do some literature, you find out that more than 70% of the literature sources that you'll be seeing will be citing Caucasian or Asian sources, and that's where their cohort comes from. But you are going to use these drugs in Black people, in Africans, not African Americans, now in Africans based on the continent of Africa. And we know the role epigenetics can play in drugs. We know the role that genetic makeup also can play in drugs. So given that existing racial difference and population, most of the studies need to be conducted here before these particular agents are allowed to be taken into the general circulation. And so that trend where the large pool of individuals with drug hypersensitivities in Africa are neglected is actually a disservice to us and to other developing parts of the world. Thank you for listening.
Yes, I must appreciate the participants, the person of Professor Olushola Aremo. I also appreciate the presence of Professor Olaiwola from the University of Obafemi Awolowo University, Ilefe, Dr. Demola Suleiman. Your presence is felt, and Professor Yemisi Kunle. You are also appreciated, ma'am, for the attention you have given us. Dr. Adekoju and Professor Auriagba, I also appreciate your presence, and every other person, all the distinguished personalities have graced our meeting. The floor is open for questions and contributions as the case may be. So we will appreciate if you raise your hand if you have questions, or you can send message. You can send message. Okay. We also appreciate. I want to call on the Modupe Ali to ask the question.
You have to unmute her. Okay, I can't hear her. I can't hear her.
No, we just have to pursue and then provide some of the food. You understand? Okay? Because a patient can even be wielding, like even as we are talking here now, there are not less than 4 or 5 doctors here. You understand? And then you may need to just do a quick examination. You can't be taking the patient to another place. Even if this is an office, at least this examination couch.
Okay, thank you very much, Dr. Amali, for the presentation.
Yes, ma'am.
You are talking to a man, actually. Okay. I want your opinion and views on this, because I've always had the issue with the issue of type A ADR.
Okay.
That is due, and when you relate it to the side effects, they said it's dose related, but based on the classical definition of an ADR as being a reaction that is harmful and is at the dose normally used for treatment, therapy, and prophylaxis or medication of physiological function. So can we actually say now that type A is like an exaggerated side effect? So then if it's an exaggerated side effect, so why do you now term it as an ADR in the first place? I just want to view, basically. Thank you.
Now, sorry, Pharmacist Amali, before you take that question, I want to combine it with what Professor Olaiwola sent me, that you should try and explain to the audience the mechanism of adverse drug reaction happening with digoxin, even when taken at the therapeutic dose. I think it's more or less related to what Dr. Soyemi just asked now. So I think I would like you to take the two together. And then finally, what's the feasibility, a question from Dr. Ademola Suleiman, and the practicability of using this predictive method, those methods you mentioned in our institutions and research institutions.
Okay, so I'll start with the first one. So we said type A reactions are exaggerated responses that are supposed to be, should I say, exaggerated responses of the side effects of a particular drug. And what the questionnaire asked me was that if this, now one thing I want you to understand is this. That's why the whole paradigm, if you remember, when I started, I said something about idiosyncratic reactions and why I don't generally agree with this particular concept of idiosyncrasy. Because now we actually know practically everything about onset of adverse drug reactions. In fact, you can know the exact dose at which this particular adverse drug reaction occurs. So for example, you're taking acetyl salicylic acid, or you're taking paracetamol, or whatever drug it is you're taking, right? And at the recommended dose, you're seeing an adverse drug reaction. Experimentally, if you carry that particular molecule, it will take that particular molecule into the lab and into a closed system. You'll find out that you will get to a certain dose. If you taper the dose gradually from 500, if you taper it down, you'll find out that you will get to a certain dose where those manifestations that are being expressed at that 500 or 1000 milligrams will no longer be there. You may retain the therapeutic effect of that particular agent, you get, but at that particular dose, you will not see the adverse effect being sustained. You understand? So if you remember also, what I said was these are the general definitions that I'm giving you based on general understanding. Because if I delve, or if we decide to delve into all these individual parameters, we don't have enough time to look into all of that. So while that is acceptable, if we go further, you'll find out that you can break down drug hypersensitivity reactions, whichever one, as far as it is linked to the immune system. You will find out that you can break it down to know the exact component of the immune system it affects, the dose at which this occurs, as long as it has to do with your B cells, T cells, and other, what do you call them, immune components. You may see some differences if you're now looking generally at adverse drug reactions. For example, somebody takes an antacid and is constipated. Those kind of issues. That one is quite different from when you're looking at drug hypersensitivity reactions. Maybe we can see and talk one-on-one on this if you are around this part, but it's a lot deeper than looking at it from the surface. Because even currently, we now have regenerative medicine therapies, and they have their own way of assessment, because the adverse drug reactions that are prevalent in those therapies are even more severe than the adverse reactions that we're currently witnessing, leading even up to the development of tumors and all that. So that one is even another sphere altogether.
On the question of the issue of digoxin.
Yes. So on the question of the issue of digoxin, yes, sir. That is from Professor Olaiwola, who was my supervisor or my teacher in IFE, and it's an honor to be answering a question from him. For digoxin, it is more of a cardiovascular effect that is seen. And so in the prism of this, our discussion, I don't think I'll be able to do justice to that hypersensitivity effect. Sorry, that adverse effect, not hypersensitivity, that is being observed there. But it has to do with the cardiovascular system. For now, I think I'll have to look that up and delve into that a bit more. I won't have the answers for that right now.
Thank you very much. I would like
there's another question that I would like to also attend to from Abdusalam Ayodeji. And the question is, in the cases of multiple drug hypersensitivity reaction, how do fixed combination therapies like sulfamethoxazole, trimethoprim, and piperacillin tazobactam affect these? What's the last part?
How does fixed combination therapy of this affect the assays used in characterization?
Okay, how? Yes, so if somebody is taking that particular combination, which is common nowadays, you have a lot of combination drugs for ease of administration, cost, and all that. So it's a welcome development. All those combinations, you find out that from the test that we carried out previously, and which I was fortunate to be a part of, you find out that it is only one of these components that is usually responsible for the adverse drug reactions that you see. So if you have an individual that has taken, for example, piperacillin tazobactam and presents with an adverse drug reaction, the first thing that you do is you do the physical assessment, you see that all the signs are there. Then once you extract the individual's cells, you isolate the immune cells, the T cells and the B cells, and then you now get standard piperacillin and tazobactam preparations. And then you now test the cells separately and see which particular chemical compound or drug will be responsible for the proliferation you see, so that you can actually pinpoint the actual compound that is responsible for that. If you don't get it with the two, then you can try a combination of the compounds and you will see most likely a response. If you don't get one, then it means that the reaction you're seeing is due to another causative organism, or sorry, causative compound, and not that particular agent. So that's how that can be done.
Thank you very much. I will call on Dr. Mrs. William. I can see you have been raising your hand all this while. Please, your opinion, your question or observation, as the case may be. Dr. Mrs. William. Please unmute yourself, ma'am. Dr. Mrs. William, can you unmute yourself so that we can hear you?
The microphone at the bottom there, just click on it.
Yeah.
Okay.
You have to enable me to be unmuted.
Okay, thank you very much. You are free.
Can I speak now?
Yes, ma'am.
We want to appreciate Dr. Amali. I didn't expect anything less than what he has just done. He has made us proud in the Faculty of Pharmaceutical Sciences in Ilorin. The thing that comes to my mind is that you already mentioned the challenge of not having the equipment, the enabling environment to carry out the molecular prediction and characterization of these ADRs. That's why most of the clinical trials, tests are done abroad and the drugs are dumped on us. What is the way forward?
Okay. Sorry, after I answer her, go to the question. There's somebody that wants to respond.
I know. I've answered.
Thank you for that question. Now, I'll tell you what I met on ground and what I've tried to do and what I hope can be done. Now, when I was faced with the issue of how can this study be carried out here, I have traversed about 5 institutions. Some institutions have the equipment, they don't have the skilled manpower, and you find out that they have parts of the equipment and not the whole part. So it's like buying a car, you don't buy an engine, or you buy a car and you leave out the ignition and all those kind of things. So the equipment is there, it's not functional. But for these assays to be carried out, there are some key components that need to be available. And the first thing is cell culture. You need to be able to culture cells. And for an individual, it's a lot of money. Getting grants to set that up here is a bit tricky because the drug companies will prefer us to remain the way we are so that they will continue dumping stuff on us. So if they know the direction you are going, they will be a bit dodgy. All they just want from you is to generate samples from your cohort and send to them to use for testing so that they can run whatever they want to run with it. So what we have tried to do at the Department of Pharmaceutical Microbiology and Biotechnology, which I tried to start and which the questionnaire has actually continued, is we have tried to start the installation of a cell culture suite. And not only cell culture, cell storage, where we can maintain cells for long periods of time in a cold storage facility. I'm happy to say we have gotten the cell culture hoods, we've gotten the cell culture CO2 chambers, we have the freezers, and the main problem we had was power supply. We've been able to install the inverters, and then Corona now struck. So we are getting ready to take off at not snail speed, but at rocket speed. So that's what we're doing on our own, just little on our own, you know, and we hope that those in authority will pay more attention to all these kind of things so that they can contribute to us so that we can grow, because it's not rocket science, it's things that are done everywhere.
There's a question from Mrs. Elegbede, so that
yes.
Is science looking at the direction of prevention of ADR?
Yes. Now, science is looking at that direction. Hello? Okay. Science is looking at that direction. When you say prevention is like you're leading them into a trap. You get. So you can't prevent ADRs, but you can predict to the extent of prevention, so that rather than being reactive, you are going to be proactive. You don't have to wait. You know, you are always ahead, so that you know that this may occur, this may occur, this may occur. So you are always a step ahead, so that whenever it occurs, you will not prevent it, maybe, but you'll be able to reduce the number of adverse drug reactions to the barest minimum. So that's where they are trying to head.
Another question before we entertain I will delay is on solil from solil. Is there any positive or advantages in adverse drug reactions and hypersensitivity? Can they be put into use, positive use?
Yes. So currently, there is that's why in Africa it's a bit difficult for us. Hello? Yes. So currently for allopurinol in the UK, before you are administered allopurinol, you undergo a HLA. There's a test you undergo. If you express that particular marker, HLA marker, you are not administered the drug, because they have found out that from the bank of cells that express that marker, even naive individuals that have never taken that drug before, because hypersensitivity reactions in most cases are delayed. So they are delayed hypersensitivity reactions. But this one is a naive individual, never seen the drug, the cells have been collected, but expresses that phenotype. Once you administer that particular drug, it starts blowing up cytokines and all sort of markers of drug hypersensitivity. So that test is a priority. But here we just administer anyhow. So there is actual real-time applications of this particular kind of science. In fact, labs now in the UK, they make money from this. I know a particular lab where drugs are sent from all over the world for them to run and test, because they have these banks, they have the samples, you understand? So people don't have to go around. They have samples of all the HLA phenotypes, because it's a genetics facility, biogenetics and all that, by Professor Munir Mohammed. He's in charge of that institute. They have thousands of cells. So all you need to do, you want people that express a particular phenotype, you send your drug to them, they test it against that phenotype to see if it's a general problem, and then they can know whether to pull it out or write it as an indication. Because not too many people will express that phenotype, and if it's effective, why deprive the general population from that? Why not just that small subset? So that's it. So by the time investigation or science goes on, you find these markers with different disease conditions. And
this also brings us to another question on the pharmacogenetics. That is, when you look at, is there any relationship between adverse drug reactions and hypersensitivity reactions and pharmacogenetics? And I don't want you to go deeper in relating.
Okay. Yes. So now, as I said, one of the factors that are generally, or let me say, that play a role in the development of adverse drug reactions is ethnicity. They related it to human leukocyte antigens and the presence of the genetic pool, varying genetic pools in different individuals. Now, we know that environment influences, the onset of adverse drug reactions. We know that race influences the onset of adverse drug reactions in specific individuals, because depending on where you are brought up, your genetic pool will be different from another person. That is the first step. That is epigenetics at play. Then when you are looking at pure genetics, races have been shown to express different traits to a higher level. Some are, we know that the Asians are fast inducers of alcohol. Understand? They actually reduce it very fast and all that. Their liver, their microsomal enzymes. And basically, the same thing is applicable with drugs. So the Han Chinese, for example, express some of these HLA markers more than others. That's where the allopurinol issue now started from. Abacavir, the issue with abacavir was seen in Caucasians. But when you come to Africa, of course, we are, should I say, a naive continent when it comes to this type of studies. You don't see that kind of, should I say, characterization of the gene pool of the African. So we are only borrowing what they are giving us to do whatever it is that we're doing here. Genetics plays a large role. It may even play a larger role than what we currently know, because every day they are finding out more and more and more and more things, you know. So what we have seen now is just, should I say, a small bit of what is possible. And the limitations basically is because of the cost of this type of studies. To run one HLA phenotyping is about $100, yeah, by $100, yeah. But you now find the people who do this in large quantity can afford to run for $20, $30, $40 because they are sending 2,000 samples. Now convert that to Naira. That one can run a whole faculty in Nigeria, or a whole department in a hospital. Nobody will give you that kind of money for funds. So we need people that are ready, you know, to invest in science, in healthcare, that are genuinely concerned, you know, in all this.
Thank you. Dr. Awodele Olufunsho, you have the floor. I think you have been willing to make a comment. Awodele Olufunsho. Dr. Olufunsho, you can unmute.
Yeah. Good afternoon, sir.
Yeah, you're welcome.
Yeah. Let me congratulate Dr. Amali for making that brilliant presentation.
Thank you, sir.
I want to just respond to Francis Shuemi's question,
Yes, sir.
and also talk briefly on the question of relation between adverse drug reaction and pharmacogenomics. Yeah, let me start with the later. Well, it's been known that 7% of adverse drug reactions are idiosyncratic in nature. And as we all know, idiosyncratic reactions are basically mediated by immune system and genetic makeup. And I just give one example of reactions to primacoin, you know, which is subject to expression of glucose 6-phosphate dehydrogenase, you know, leading to hemolysis. And you also have example of slow acetylator, fast acetylator to isoniazide. So there's a very perfect, great connection between adverse drug reaction and pharmacogenomics. And that's why all over the world now, it's been advocated that people must have their genetic profile mapped out before you know the appropriate drugs to prescribe, so as to prevent adverse drug reaction. On the other side of Francis Shuemi's question, yeah, it's a very brilliant question. That why do we classify that an exaggerated side effect? The basic fact is that adverse drug reaction is an undesirable reaction, which make it different from side effects. And when you have exaggerated side effect, it becomes undesirable. It becomes severe on the patient. For example, exaggerated hypoglycemia, as a result of the use of oral hypoglycemic agents. It's become unwanted. It becomes unintended. So based on that fact, is classified as adverse drug reaction, not just side effect, because at that level of exaggeration, it becomes unwanted, it becomes undesirable on the patient side. And the other one have been properly explained by Dr. Amali, and I say thank you for the opportunity to speak.
Yeah. Thank you very much. Once again, I think there is a question from Dr. Afsad Additional. We want to know if it is workable or some claims that is, they are taking ARV, that he discovered that most of these patients were weaning off some of their drugs as a result of adverse drug reaction. That is it possible or workable to put this patient on different doses of antiretroviral just on trial before they eventually, enroll into the antiretroviral therapy?
Thank you. Yes, I would first of all like to thank Professor Awodele for his contribution. That's my very senior colleague. Thank you, sir. Now, for antiretrovirals, I saw the question some time ago. Yes, it's possible if we can take, because just like sensitivity studies, in quotes, if we can have a lab where we can assess the level that these mediators of hypersensitivity are now released in quantities that will lead to deleterious effects. Once we can assess that, then it will be possible for us to taper the dose. But you have to be careful also that that particular dose at which the mediators are not released is also a dose that is strong enough to be effective, that can be able to either kill or inhibit the virus in that situation. It is also difficult, particularly for antiretrovirals, because it's difficult to get their cells to survive for long periods outside in an in vitro system. It's quite tricky, and then the environment that they need to be worked in also is quite tricky. But I'm not saying it can't be done, but for now, the substitution of the dose of the drug, is the most feasible way forward. And clinically, I believe most of us here are clinicians and pharmacists. We know that when you're looking at management of a patient, the first thing that comes to mind is the fact that the risk-benefit ratio. Is the adverse reaction going to kill the person? If it's not going to kill the person, the virus is definitely going to kill the person. So if the individual can withstand the adverse effects that are manifested, then the best option may be just to continue with that particular drug in the worst case scenario. But it's possible to get a dose level that will be.
Thank you. On the last note, I will call on Dr. Ademola Suleiman for your contribution. We appreciate your patience all this while. Dr. Ademola Suleiman, you have the floor. Yes, I mean Dr. Ademola Suleiman. No. Dr. Ademola Suleiman, are you there?
Yes.
Yeah, thank you.
Can you hear me now?
You have the floor, sir.
Yes, let me start by thanking the presenter. Let me thank him again, and let me thank him again, because he has actually given me a lead on one of the research I'm doing, in which I know one of the objectives that I need to have. I have been running away from the ABR, but I think I have a way out of it now. And I would have loved to have asked this question before Professor Awodele speaks, but I think he can also make a contribution. Someone did ask about the issue of multiple drugs, like sulfadoxine, prometamine, and how do you do the assessments and characterization? And I noticed that the response given by the presenter, he actually, let me quote him, that most time it is one of the drugs that actually responsible for it. But we also know that this combination at times also there is a relationship The relationship could be additive. It could also be potentiation, or it can also be synergism. In such a situation, is the predictive still the same that because the outcome in case of potentiation or synergism, it is not in a summative means. So how do you do the characterization and the assessment? Thank you, sir. And thank you again for the lecture.
Okay. Maybe I'll start answering from this perspective. The fact is that one of them, most times, 99% of the time, let me say, is one of them that's responsible for the adverse drug reaction you see. Now, whether the effect you're seeing is additive is another issue entirely. The fact remains that once you take the causative agent out of the mix, even if it is an additive effect, the fact remains that the agent that is enhanced to carry out that activity is no longer there. So you don't see any manifestations of that particular adverse drug reaction or drug hypersensitivity reaction again. Okay, and then you can now use another substitute. The ideal approach is you use both of, or if you have a combination of a drug with a combination of three compounds, you either use one of them, that's each one individually, and then you use combinations of them. So for example, as I said earlier, if you have sulfadoxine and pyrimethamine, you have your sulfadoxine test, your pyrimethamine test, and then you now have a combination of the two and see which one would produce the effect that you're seeing. There you can actually quantify and see if the adverse drug reaction is either additive or it's based on the same level with that you see with the individual compound. Because most of the time, the synergistic effect you're seeing is directly related to the therapeutic effect and not necessarily to the manifestation of adverse drug reactions.
Thank you very much, Dr. Amali, for the message so far. I would like to call on Mr. Eto to also put own contribution for to give us a photo of thank.
Just unmute. Unmute her. She's permitted.
Okay, now.
Mr. Eto, you have the floor.
Okay. Well. For over 1 hour, we have a lecturer here, Dr. Mohammed Amali from Ilorin, doing justice to what he has known for a long time. And we want to appreciate you for calling upon you, and you answered us. You have spent most of your time with us here today, and we appreciate it. On behalf of our head of department here, pharmacist Abdurrahman Afolabi, and all the pharmacists in pharmacy department here in teaching hospital, we want to thank you and appreciate you for coming. You have done many things for us here today, and we cannot forget it. You have participated with us during our research on adverse drug reaction in our medical world in 2017, and you really did what we wanted that time. So we thank you all the same for all this, and when we call upon you next time, we know you will answer us. Thank you for your time, and we thank everybody for listening. May God bless us all.
Thank you.
Thank you very much. I once again appreciate everyone for your rapt attention. We are going to send this message to our YouTube channel Then we send to your email address. If you are interested in these slides, you can send your email in so that we can forward it to your various email address. On behalf of the head of department of Pharmaceutical Services, University of Ilorin Teaching Hospital, pharmacist Abdurrahman Afolabi, and the entire staff of pharmacy department of the same unit, of the same institution, I thank all of you, and I appreciate all of you. Thank you very much. We have come to the end of this presentation today. May all God be with all of us. Thank you very much.